Let's keep thinking about origin of life stories. Welcome to Critical Thinking Scan, where we look at how you can think about any faith-challenging message and arrive at a biblical, logical conclusion yourself. I'm Patricia Engler, and today let's continue part 2 of 2 videos about life's origins. Even if we could miraculously achieve an amino acid sequence that made a functional protein, steps 5 to 7 in evolving life would require arranging the protein into the right secondary, tertiary, and quaternary levels of 3D structure. That includes folding the protein the right way without the usual help from other proteins. But even if we could do all of that, that would just leave us with one protein. As of 2019, the smallest synthetic bacterial genome consisted of 473 genes, showing just how many unique proteins must work together to form even the simplest cell. And proteins don't store information for cell maintenance and replication. That's where integration with DNA and RNA comes in, which is step 8. But as a 2018 paper understated, it is unclear how the initial relationships between RNA and proteins came into being, bearing in mind that RNA needs proteins to replicate and proteins need RNA to be coded, a problem known as chicken and egg dilemma. One proposed solution is the RNA world hypothesis, which suggests RNA existed first because it can store information, replicate itself, and perform some biological work. Yet, as one textbook admitted, from a purely chemical standpoint, it is difficult to imagine how long RNA molecules could be formed initially by purely non-enzymatic means. That's because RNA requires proteins to form under real-world conditions. Some researchers suggest that RNA must have come from simpler compounds called pre-RNA, but the textbook observed, "We do not have any remnants of these compounds in present-day cells, nor do such compounds leave fossil records." In other words, there's absolutely no evidence for pre-RNA. It's an assumption requiring blind faith. RNA is also less stable than DNA, and recent studies have pointed out numerous other problems with the RNA world scenario. For example, it doesn't explain the genetic code's origin, which would be step 9. One paper wrote, "Such a rise from RNA to cell-based life would have required an out-of-the-blue appearance of an AARS-like protein, those link amino acids to tRNAs, that worked even better than its adapted RNA counterpart. That extremely unlikely event would have needed to happen not just once, but multiple times. Once for every amino acid in the existing gene-protein code. It just doesn't make sense." So, various researchers propose another just-so story called the RNA peptide world, where RNA and precursor proteins slowly evolve together like a chicken and egg, forming simultaneously without outside intervention or foresight. The story goes, the evolution of peptides to proteins would occur from small motif to domains and finally folded proteins. Those proteins would have, in theory, evolved increasingly complex functions. But remember, even the simplest engineered cell requires 437 functional genes and proteins working together. There's not just one chicken-egg paradox in cells, but many, as a linked article documents. And even if against all odds one cell could appear naturally, that still leaves subsequent problems of gaining new information for evolving more complex cells, evolving multicellularity, and evolving new kinds of organisms. Ultimately, hypotheses that life evolved from non-life aren't conclusions you'd reach by starting with observational science. They result from starting with naturalistic presuppositions and trying to wrestle science into them. So, when you encounter origin of life claims, always ask, what's observational science and what's historical speculation? Assumptions about early Earth conditions shape studies like the Miller-Urey experiment, which observe certain compounds forming under controlled conditions. But statements about how those observations relate to life's origins, those are historical science. And a close look at other observational facts typically reveals why the experimental conditions would cause problems for life's origins under real-world circumstances. An entire Nature Communications article exposes how heavily such experiments rely on intelligent human intervention. And keep in mind the multiple interdependent steps required for life to exist. Besides, even if against all odds, researchers showed that life, or even one protein, could possibly have evolved naturalistically, that wouldn't mean that it did happen that way, just that it hypothetically could have. Confusing could with did is the fallacy of appeal to possibility. There's no way to certainly demonstrate what happened at life's beginnings because we weren't there to see it. But in a biblical worldview, Genesis reveals God's infallible eyewitness testimony of life's origins. For more on how to think critically about faith-challenging messages, you can access my other CT Scan episodes packed with tactics, tips, and tools that helped me as a Christian student at Secular University. Thank you for watching. Hey, it's Patricia. Just wanted to let you know that if you like these videos, a free, easy way to help Answers in Genesis Canada produce more content and equip more people to defend their faith is to hit that like button, subscribe to our channel, click the bell, and of course share these resources. I know you might hear that kind of thing a lot, but the reason these actions are so important is they inform social media algorithms to help these videos reach more people who can benefit from them. And that's especially helpful because advertising is super expensive. But this way, even media platforms which are often unfriendly towards biblical content become tools to promote gospel outreach for free. And if you're on board to share this message of biblical authority and want to give, you can also make a one-time donation or become a monthly supporter by clicking the link below. Thank you so much.